All right, we're talking about digestion. So carbohydrate digestion. We just spent a whole bunch of time talking about the mouth and the esophagus and the stomach and the small intestine. We haven't really talked about the colon or anything else yet, but we're going to switch really quick because of all of the chemical digestion that occurs in the small intestine. And we're going to talk about starches. So starch, like potatoes, French fries, you ever put a French fry in your mouth and it does almost melt the center part? That is a digestible carbohydrate. We call it starch. It's from plants. It's what they keep. But cellulose is the other carbohydrate from plants, which we as humans super duper need. You can't digest it, and that's what we need. Because we're not lions, we don't have super short intestinal tracts. Our alimentary canal is like, 30 to 40 feet long. And what the fiber does, it actually brushes everything out, and it's like a housekeeping, clean it up thing. Right? So even though you don't like fiber, just get fiber because it's a primate thing. And so it's an important part of our diet. Cellulose is that's the fiber part. Okay. The starch is digested into smaller starches, really oligosaccharides. This means it's like a couple of sugars stuck together, like up to 8, not beyond that. Then that gets reduced down to like a 2 sugar molecule, a disaccharide called maltose. And then maltose gets digested down into glucose, which is like the humdinger of our respiratory system when we're talking about the respiratory system, Krebs cycle, electron transport chain, all that. And now that gets absorbed into the small intestine and distributed throughout the body. That's the blood sugar we're talking about when we talk about blood sugar. So you can go from a very complicated sugar to a very simple sugar, and then that goes straight into the bloodstream. Okay? Because we want to get carbohydrates as fast as we can and the most amount we can, carbohydrate digestion begins in the mouth with the amylase, which we see lots and lots of. We see amylase again later on from the pancreas. It denatures quickly those starches down. And right, so most of your dietary starch is actually digested before it reaches the small intestine, and some of it even absorbed. Okay, that's right. Right, so upon reaching the small intestine, your pancreas is also going to push out amylase as well, right? Brush border enzymes are going to continue breaking down into littler ones like we just talked about, into the maltose and then the monosaccharides. And then here is that image of the pancreas itself. Looks like a J, right? So again, when you look at this pancreas, I want you to notice there are 2 exits from the pancreas. There's one right here that we're looking, there's one that we combine with the bile duct, but then there's one that always kind of drips out pancreatic secretions. It's always kind of going to have a little amylase in there. We want to catch any kind of leftover starches. We want to get every little bit we can. Because glucose in a natural environment, meaning like if humans were 12,000 years ago back in the original environment kind of thing, not in 2021 first world, glucose is so hard to come by. It's like in berries in the springtime, maybe any apples you come across. It's very rare, right? Super valuable stuff. 80% of your absorbed sugar is glucose. It's taken up by the SGLT, sodium glucose transport proteins, really transport. It is going to be transported out into the cell very quickly by facilitated diffusion, right? So very quickly. It actually just kind of gets absorbed in, how do I want to say this? It's like essentially it just gets, oh my gosh, I can't believe I can't think of the word. Resuspended. Oh, there it is. Resuspended in the cells that are absorbing it, and then gets pushed back out with water, right? And so water is going to carry most of it, and it's dissolved in that water, and that's how it's getting to be moved quickly into and through the bloodstream. Fructose and glucose and galactose are some different types of like very small sugars, right? Like smallest you can do. Some only have like 5 carbons, others 6, so stuff like that. They're just different arrangements for the most part. They're going to get converted generally the glucose, because that's what our cellular metabolism, right? Electron transport chain, Krebs cycle, glycolysis, that's where we're at with that. That's what we're going to use. It gets absorbed readily into the blood in the villus, in the small intestine very quickly. And then the hepatic portal system, that's why when you eat sugar right away, it goes straight to your liver, because the first stop on the absorption route after your stomach, it takes it right up through the portal system to your liver, where it gets observed, see how much you have in there, and then gets processed and sent back out through vena cava. So we'll talk about lactose intolerance real quick, because it's an important human thing. So about 12 to 10,000 years ago, it ended up being that for whatever reason, at least 3 separate populations, 2 in Europe and 1 in Africa, started losing their ability to turn off their ability to still feed on lactose. And here's the thought. So until you're about 5 years old-ish in humans, maybe older, you can digest lactose normally. And so then what happens in every mammal, right? Like all mammals do it, like kittens when they're little, like deer, like they're all, have mother's milk. But what makes them go from something delicious like milk, which has fat and sugar and everything you need, to eating if you're an elk, grass, if you're a cat, good heavens, the things that cats eat. My cats had cockroaches, for heaven's sake. You know what I mean? It's just not good. They still liked milk, but every time they drank it, they threw up. Have you ever noticed that? If you give a cat milk, it's like, oh, good kitty. And then 20 minutes later, puke pile. Okay. That actually happens and is supposed to happen with all mammals. Okay. And humans are included. And so what ends up happening is this: it forces you off of nursing. It forces you into the environment, because heaven forbid somebody becomes a mother and then however many offspring they have just continue to nurse for the rest of their life, right? There has to be a driving force. And so it's thought that this is what it is, right? This inability at a certain age to digest lactose. Well, humans have the same set of genes. And if you can eat lactose and not have diarrhea or stomach issues afterwards, then basically you've had your gene that shuts it off naturally at a younger age is broken. Okay? So now it's always constitutively on so you can digest lactose. And so these 3 independent populations that they found, around 10 to 15,000 years ago, prospered. And these individuals who could digest lactose, took off, right? They had huge amount of descendants relative to the people who had a harder time. We don't know why. It might have been something like there was a famine. The subsistence strategy might have had to shift to where they were dependent on milk from herd animals, which they had at the time, and that allowed them to survive. So it's hard to know. But most of the population on the planet is actually lactose intolerant. So that's something that arrived in Africa. So I don't like what they say here. They say occurs in many people, and they're like white people and black people and Mediterranean and all this other stuff. But the truth is, it happens in whoever carries those genes from those original populations. And so you don't really know what color their skin is always going to be. Okay? Especially in places like the United States, which has a ton of admixture. And we live in New Mexico, so that's really mixed, right? There's people in my family who are lactose intolerant, and then this brother of them is not, right? So you get this level of mixing because it's just following gene flow, okay? So it's kind of an interesting thing that happened back then, and it's very prevalent now. It's something that took off, and it added value to people's subsistence strategy. So they say nearly all of Asian descent, but then you look at Mongolia, and those populations are highly dependent on horse milk, right? And very, very successful. And obviously Genghis Khan is very successful. Not great always, however history shakes out, but still it is what it is. So it kind of So what you need is you need the ability to produce one enzyme, and it is lactase. So should you be able to continue to produce lactase beyond like the age of 5 or into adulthood, then yeah, you have the mutation. It shuts down, and it shuts down your original inhibition for it. It's pretty interesting stuff. And the bacteria go crazy over lactose, right? It's a really easily digestible sugar for them. And so it causes like some gut issues, and it's just, it's diarrhea city. Okay. Protein digestion. Amino acids absorbed into the small intestine come from 3 different sources. You're going to eat it. You're going to also eat your own enzymes and proteins, actually. You're going to digest your own cells. Okay. And then also not mentioned is anything from the bacteria that's living in your gut, which has a significant weight value to your body weight. You'll also be digesting those ones as well. Okay. So about 30 grams a day. Exogenous is the rest. 30 grams a day. That's like the size of like a hamster, maybe like a mouse. If you just want to put it into perspective there. That's not helpful for some people, sorry. Okay, so proteases degrade protein. Peptidases are some that are very specific. They like to work in acidic environments. They have different environments acid-wise, okay? Pepsin hydrolyzes a peptide bond. So go back and look at hydrolysis. And what it means is like you take a little piece of water and you split the water into an OH group and a hydrogen, and you like smack down and you lyse, or you break open a molecule, you break it apart and you cap it with those 2 parts. So go back to, your biology, and so there's that. That's what it basically means. Okay. Here's protein absorption. So there's not like really chemical digestion. There is some amylase-wise, but we're talking about protein digestion. You have to really wait until you can get that high pH value to where we're starting in this case. Continues on into the small intestine. Pepsin is one of the beginnings, and it needs a higher pH. It needs 8 or alkaline. Okay, so different pHs for that. Trypsin digesting starts happening as well. More and more hydrolysis, remember, breaking apart of these proteins using a water. Okay. Oligopeptides. Oligo means like a chain of many. That's all oligo means. Not like crazy huge, but like, 10, 8, something like that usually. So it's taken apart a single amino acid at a time. So it just like pops one off, pops off like little beads. Carboxypeptidase removes amino acids from the end of chains. Aminopeptidase takes the amine group off. Dipeptidase just splits different, proteins that are only 2 monomers long. So there's different types of, I don't want to say ases, but enzymes. Yes, anytime you see an ase, that's an enzyme. And they do just different functional things. They're functional, Okay, and you don't need to know this. We have it. It's part of the book. There it is. Actions. Okay. Brush border enzymes at the ends of the cells on their microvilli are finishing up their tasks. They need sodium to move the single amino acids that have been digested from the bigger proteins into the cells so they can like use it as a nutrient and move it around the rest of the body, right? So there's that. That's what a co-transporter means. Like it's required to transport together into the epithelial cells. So then we also use facilitated diffusion to move it out into the bloodstream. So it's like a lower energy process, lower. So there's that. Okay, here's kind of an idea of what is happening. It just comes here in the microvilli, and it actually crosses the cell, and then it gets secreted directly into the capillaries, and that's the absorptive process, and it needs single amino acids for it, right? So not huge proteins, single amino acids, the building blocks. The hydrophobicity of lipids makes it hard to digest fats, right? Because there's a lot of water in your guts, there's a lot of water in you. And so, in order to get fats, which we need to make things like the borders of our cells, right? So, our cell membranes and things like cholesterols and all this other stuff has to be digested and emulsified very specifically by lipases. We talked about the lingual lipase, which starts in the salivary glands in the mouth. So we need to kind of, we don't have a high rate of it. So only like maybe 1 in 10 or so percent, or 1 in 10 are going to be digested by the time you get to the duodenum. So not a lot. Emulsification is going to be what happens primarily in the small intestine. And then that's, remember, the breaking down of the fat droplets. Okay. So things that break it down: bile salts, lectin, just moving it around. What we want is we want those hydrophobic tails to flip out so that we can grab onto them, digest them, and start absorbing them. Great. So monoglycerides and other lipids are going to have to be reduced to these tiny little droplets. And we call these the tiniest droplets you can have are called micelles. And it's like individual tiny fat molecules, and they wrap around each other, and sometimes a little bit of water, and it's a micelle. So these are going to be made in the liver. They're so small, they're only 20 to 40 little molecules long for acid. And that acid is going to work on the charged end of the lipids to kind of pull them apart. Okay. So the micelles are going to be pushed into the bile, move down the bile duct to the duodenum. They're going to be used to do things like pull in fat-soluble vitamins like vitamin D and vitamin A, for example. Cholesterols that can be used, that we use later on after they've been digested to reproduce steroids and other hormones, for example. They also help transport the lipids to the surface of the intestinal cells so they can be absorbed. The nice thing about these little micelles is they can actually pass directly through the plasma membrane because the plasma membrane is fat itself. So it's nice in terms of cargo. Okay, here's kind of what it looks like. So tiny. Look at these tiny little lipases. Here's the acidic part right up here. Here's the food particle that it's surrounded, right? And so it's going to be able to just diffuse right onto the cell once it docks. So once it's in the cell, individual fatty acids that kind of get broken apart, they can be moved directly into the smooth ER, and they start getting processed into what other kind of secreted elements that cell is going to process, put together, and excrete through the smooth ER and the Golgi body. Right? Yeah. After it moves through the Golgi complex, if you remember way back in biology, it gets put into a secretory vessel. And so that's like another micelle essentially that's going to have that final finished product. Maybe it's made testosterone or estrogen or hormone, and it pushes it out to the surface of the cell. Okay. Oh, my bad. Let me get back here. Out to the cell. So, let's see. So it's going to be taken into the lymph, right? Little lacteals, where the terminal ends of like almost capillaries of the lymphatic system. Fats are absorbed into the lymphatic system. Just kind of to put that in there. I forgot to mention it. All right. So let's get into the large intestine next.